Welcome. We will wait for 2 minutes for the participants to join, and then we will begin the seminar. Thank you. Let's wait for 1 more minute and then we can begin. Welcome to the EuroCC-Turkey seminar series. The EuroCC-Turkey team has organized weekly seminars since December 2020 to disseminate HPC among academia and SMEs. The interdisciplinary seminar series covers parallel programming, GPU programming, big data, machine learning, computational fluid dynamics, and materials modeling. You can follow our events on our Indica webpage and watch our previous seminars on our YouTube page. To learn more about the EuroCC project, please visit the link in the comment section or the chat section. Today's talk brings together computational biology and deep learning domains. Our speaker is Dr. Ercument Cicek. Dr. Cicek is an assistant professor at the Computer Engineering Department of Bilkent University and an adjunct faculty member at the Computational Biology Department of the School of Computer Science at Carnegie Mellon University. He was a Lane Fellow in Computational Biology at Carnegie Mellon University. He received his PhD degree in computer science from Case Western Reserve University in 2013. Dr. Cicek, the floor is yours.
Thank you very much. First, thank you for the kind invitation. Today, I'm going to talk about one of the projects that we were working on for the last 2 years with the Alkan Lab at Bilkent University. And so we are going to talk about copy number variants, detecting them on the genome, and polishing them with a deep learning approach. So this work has just been accepted to Genome Research Journal. So I think like within the last 2 weeks, we'll be in the June issue. So it's still on bioRxiv. So if you wanna take a look at the paper itself, you can take a picture of the QR code on the right button. And so let's start first. And kind of a brief introduction to the sources of genetic variation in the human genome. So here on the left, you see the frequency of the variation that we observe. The most frequent variation on the genome that we observe are single nucleotide changes. So single letter changes or small deletions, small insertions onto the genome, which we see very frequently. And then, so here BP means base pair, meaning a single nucleotide. As we increase the size, but once we come to 1 kb, so 1,000 nucleotides, up to 1 million nucleotides, these changes are called copy number variants. So they are obviously much longer, and they are less frequent than single nucleotide changes, obviously. And then as we go along, once we have chromosome-wide changes, like an extra chromosome, we expect 2 chromosomes, I mean 2 copies of a single chromosome. But you might lose one, which is monosomy, and you might get an extra one, which is trisomy. These are much rarer, but a lot, they are on the order of millions of nucleotides. Now, so our focus today will be on detecting copy number variants, short CMVs. So there are multiple types of these. One is deletion. So here you see the reference genome, which we consider as the healthy human. Actually, it's not a single human, but kind of an aggregation of initially 8 healthy, or otherwise, and we consider healthy, but we don't know for sure, of course, with no obvious health problems, let's say. So this is the reference genome of that, representing a healthy human being with no obvious problems. So this portion is deleted on the genome of the person that we are considering. So that this portion is gone. So we might have novel sequence insertions, meaning, on the reference genome, we don't have that portion, but for this person, we have a sequence inserted. We might have what's called a mobile element insertion, meaning you have this piece of, DNA on some other part of the genome, on the reference genome, but this just comes back and then inserts itself in between these 2 regions, which we do not expect. We have tandem duplication. So you have a certain region on the genome. We expect to see 2 copies of this portion, 1 copy from our mother, 1 copy from the father, but we have instead an extra portion. So if this is duplicated, we will have 3 copies. So 1 on 1 chromosome. Since this is duplicated on this copy of the chromosome, we would have 2 copies, so we would have 3 copies. So this is called copy number variation. When it's tandem, it is right next to it on the genome, or you can have interspersed duplication, meaning you have that portion duplicated in some other region of the genome. So these I will show you some statistics about it, but they are important sources of genomic variation among us. What makes us different about the genomic diversity as a population and for the fitness of the population. These are really, really important, but if they come across certain important genes, which we call conserved genes, important for the function or even the survival of the organism, then they might cause certain diseases called, autism is a very famous example. Specifically, the psychiatric disorders, we might have some portions of the genes deleted. Or for instance, the gains, so this is a deletion example, this is a gain example, you have extra copies. So that gene is extra active. You have schizophrenia or psoriasis associated with these changes. There are other types actually which are outside of the scope of today's talk. So you might have a certain region inverted, or you might have even a chromosome break apart and then gets attached to other chromosomes, which is called translocation. But as I said, this is going to be the focus of today's talk. So we want to be able to detect the copy number variations in a certain individual, potentially having a phenotype like cancer, and we want to understand the cause of cancer, so we can go for the personalized treatment. So copy number variants. So this is a paper from 2015, a Nature Reviews Genetics paper. The 5 to 10% of the human genome contributes to copy number variation. So this paper basically considers 2.4 million CMVs detected in 55 studies and just looks at the statistics. So they find 5 to 10% of the genome has copy number variants. They also, as I said, it's an important source of genetic diversity. I mean, they find that more than 100 genes can be removed with no phenotypic consequences, meaning you can just cut that portion of the genome from a person, it affects nothing. So it affects nothing, meaning, you know, the person can function properly, but also, you know, that's what makes us different as humans. As you see, for instance, the mean interval lengths for these regions, like 10,000 nucleotide regions, are usually copied. So this is the mean length. And thinking about it, like we have 3 billion nucleotides in the human genome. So this is a very, very short region. Actually, even if, you know, we say 5 to 10% of the human genome contributes to copy number variance, as an individual, we do not have many, but as a population, it's very common. You know, everybody has some, and then when you look at it with this study does, when you look at it in, you know, when you look at the bigger picture, as a species, we have a lot of them. Like 10% is a big number, but as a, you know, individual, you usually have a very small number of copy number variants. Now, so as I said, you know, they are important, contributes to genomic diversity, causes certain diseases, and we want to be able to detect them accurately. And now I need to talk about the technology that lets us to, you know, pinpoint these copy number variations. First, I need to talk briefly about high throughput sequencing, which is the current standard of today. So basically, if you want to, you know, some patient walks into the clinic, you want to find out which mutations they have, copy number variants in particular. What you would do is you just get a blood sample and then put it into what's called a sequencer, which is supposed to generate your full sequence of DNA, which is 3 billion nucleotide long. And if your DNA sample is a base, you expect to put it into the sequencer and to get the ways out. So like even a single string, which is 3 billion letters long. But unfortunately, that's not how the technology works. This is what the sequencer gives you. It basically breaks the ways apart. You just get the pieces. And you're supposed to put the pieces back, which is a big computational problem. You should appreciate, I think. The upside of it is that the sequencer does not give you each piece only once. It gives you redundant pieces. So if this is the genome and then you put it into the sequencer, you get, you know, short sequences of length, usually 100 to 150, depending on the technology that you use. But you get a redundant sequence that covers the same part of the genome. So this is what you would get. So so we have the technology to put them back, align them back onto what we expect, what we accept as a healthy reference genome. This is what you would get. So redundant small pieces of strings aligned onto the reference gene. Okay, so how can I use this to detect copy number variants? Now I need to talk about 2 strategies on performing this high throughput sequencing experiment. The first one is whole genome sequencing. So you basically sequence the full genome of the person, 3 billion nucleotides. And then you would have basically a read alignment like this. The second one is what's called whole exome sequencing. So in whole exome sequencing, you only sequence the parts of the genome that encode a protein, which we indicate as active part of the genome. The other parts are, in the near past, actually, we considered the other parts as the junk DNA. Apparently, it is not. The rest of the DNA is responsible for regulation of transcription, production of the mRNAs and genes and the proteins, folding of the DNA. So they are also not junk. They are also active in the sense that enabling the transcription, the expression of the DNA. So what you do is you have certain parts of the genome that that encode a protein. So you only sequence those parts. So as you see that you have some regions that are not covered by these reads. So you can do either of these. So, and I will talk about the advantages and disadvantages of those. But first, once either one of those you perform, you align your reads onto the sequence parts of the genome. This is what you obtain. You basically obtain a read depth signal, if you will. So, if you have more redundant reads, you have more depth here. So this is the number of reads that are aligned to that part of the genome. If you have a copy number variation like a deletion, then you expect that that region is not covered by reads because that person did not have those reads, and you couldn't align them back to the genome. You see like a sink, a depletion of the signal in this region. And if you have a good algorithm, basically it can say this part is deleted as a copy number variation. Or, this is what you would expect on the, the mean read depth. If you see a spike in that region, then maybe there's a duplication here because this person, if I go back, had a duplication here. So it would produce two copy, instead of a single read or, instead of 1 unit of reads, as you would expect here, they produce 2 units of reads. So once you align them back onto this single region on the reference genome, you see a large spike on that region. So you might conclude that this region has a duplication. So we performed the high throughput sequencing experiment, and then we, either whole exome or whole genome, and then we obtained right read. So, and then we are going to use this read depth to, decide if we have copy number variants in which regions or we don't have at all. So which strategy we should go for? So in the whole genome sequencing strategy, this is really good for copy number variation detection because we have uniform read depth distribution over the whole genome, even what's called a near Poisson depth coverage. So this has very nice statistical properties. Once you see an abnormality in the signal, it is almost free of noise that you can conclude with your algorithm that there's a copy number variation in that region. Whereas this is quite problematic if you do it on the whole exome sequencing data, because as you see, you have regions that are, you have gaps in the read depth signal. And this is quite noisy. The read depth is not uniform, as you see. And then due to the technology, which I'm not going to go into the details, there are batch biases. With respect to the content of the letters here, you have biases. And then the probes that you put, so you put probes into these regions to catch these reads, they have biases. So the technology introduced too many biases to on the read depth coverage that you have. So it's quite noisy. You cannot distinguish you cannot distinguish a copy number variant from the noise the technology introduces. And then we have here almost the false discovery rate reaching up to 60%, as opposed to 5% on the whole genome sequencing data. So this is pretty much useless to use in the clinic to put a diagnosis on a patient and to offer treatment. Now this is quite unfortunate because this has big advantages compared to this. This data is quite smaller. You only sequence like I think 1% of the genome, which what we consider as active. So you get a file like 3 gigs, whereas the whole genome sequencing data is around 100 gigs. And then this is quite cheaper. So this, you would expect the whole genome to cost more than $100,000 nowadays. This would be, I think, like $200 to $300, roughly speaking. And then we have, since this is cheaper, smaller, this is quite abundant currently, as opposed to whole genome sequencing data. And one other reason is, even if you find variants on the parts of the genome that are not active, it is very hard for the clinician to interpret them. What are you going to do with it? Well, you don't know the meaning of the mutation. Whereas if you find the mutations here on the exome part, meaning the active part, you can say, okay, this protein is, has a deficiency. That's why you observe cancer in this patient. So this is cheaper, more abundant, more compact. But unfortunately, due to this high false discovery rate, we cannot use this in clinic. And this is very hard to do. It takes longer, more expensive. So this is a quite big of a dilemma. I mean, we want to be able to perform copy number variation detection on whole exome sequencing data. And this is an example actually of the problem with the read depth signal. This is a real sample. What you see over here is an IGV screenshot. Here you see the chromosome 1 of that patient. And then these gray dots over here you see are the reads that we have sequenced from them and then align them back. So this is basically the read depth. Here you see the read depth information. As you see, it's quite uniform. And then here you have a spike. Here you have a spike, maybe here. Some regions are depleted a little bit, but pretty much a quite uniform signal. Whereas here you see for the same person, same sample, the whole exome sequencing version. As you see, there are a lot of gaps. You have some spikes which correspond to nothing here, and then you have just some peaks. So it's very hard to distinguish what is a copy number variation and what is basically a bias introduced by the technology. But so our goal is to be able to perform that operation here. So these are some statistics from the algorithms that do this on whole exome sequencing data, what we want to do. And so these are two benchmark papers showing the sensitivity of the methods, false discovery rate. As you see, the lowest false discovery rate is 26% with 70% recall. For the amplifications or duplications, for the deletions, it's 50% false discovery rate, 76% recall. So you see that the lowest here is 41%. So these are basically very nice attempts, but they cannot be used in clinic due to these error rates. And this is the concordance of 4 methods. This is another benchmark paper. As you see, everybody almost predicts a different copy number variation, and then they hardly agree. As you see, we have 14,000 here. This is one of the best ones we find, but it agrees with only at 700 or 800 with the other ones. So these are big problems. Even though we have many algorithms, we cannot use them for the actual purpose to help the patients. So what's our goal here? We want to be able to do copy number variation detection on whole exome sequencing data. And we want to be able to improve the performances of these algorithms using machine learning. So the core idea here is to be able to use some ground truth labels for the regions that we consider on the whole exome sequencing data. We want to use some input like the human expert input, saying, okay, we have copy number variation here and that, maybe using some other technology. So if the human expert could tell us, which regions had the copy number variance, I would just obtain the whole exome sequencing data and learn a model which just gets the whole exome sequencing signal and predicts these changes. So we would guide the algorithm with the human expert. The problem is, it is even very hard for the human experts to detect them. The best we have is by this paper with Cicek et al. From 2019. They did it on only 9 samples. So they still use algorithms, automated algorithms, 15 of them, and they get the consensus. They get some other data, some other sequencing data, and they say, okay, we have a copy number variation here, we have a copy number variation there. But if you have just this data for only 9 people, this is quite problematic. You wouldn't be able to train a neural network or any model that would learn, an association between whole exome sequencing based calls and human expert calls. So what can we do? The next best thing you can do is to use, again, automated algorithms, predictions, but obtain all the whole genome sequencing data. As I showed you, they are very accurate. So we have like 5% false discovery rate. So what I want to do now, instead of relying on a human expert, use, input the whole exome sequencing calls and get the labels, the correct, potentially more correct or more accurate labels on the whole genome sequencing data, and learn a model that would be able to bring the performance of CME calling on the whole exome data to whole genome sequencing data. So this is the key idea of our approach here. We are going to train a model that would input the whole exome sequencing read-depth signal, which is cheap, abundant, compact. And we are going to have algorithms, the algorithms that I showed you, to make calls on this data. And we are going to learn to correct those calls using the automated calls made on the whole genome sequencing data, which are much more reliable than the calls made here. So we are basically proposing a polishing algorithm, which would be correcting the calls. So you have to still rely on the whole exome sequencing callers, have them do their work, but then I will post-process them and eliminate the wrong calls or even correct the calls that they make. So how do we do it? We are able to do this thanks to this 1000 Genomes project, which is a very famous big sequencing project from early 2000s. So they sequence, I think, more than 2,500 people using many different technologies. So these are healthy people. Again, we don't know any health problems that exist with them, and they have matched whole exome sequencing and whole genome sequencing data for these people. So we obtained 802 of them. We split them 90 to 10. So we'll perform training on close to 720 samples, and then we'll do testing on the remaining 80 samples. So what we do, so for each sample, we obtained for these 5 whole exome sequencing based callers, make their calls. So these are like the best ones that we came across from the literature. So on these 812 samples, we made them make their calls using whole exome sequencing data. And now we have the whole genome sequencing data-based calls for the same samples using Seminerleri and Genomes 2 copy number variation calls, which are much more accurate than these. So for instance, Seminerleri has sensitivity from 86 to 96% and false discovery rate 3 to 20%. And then even with the data that we use, we expect this to be much lower, even though we don't have a ground truth assessment. So these are going to be our labels. These are going to be our features, and we want to learn a model. So the model we call is decont, and then this is what it does. So first we get the whole exome sequencing based read depth signal. So we have a tool called SumMambo, which calculates this read depth. And as you see, some regions are not covered because those are not the active regions. And we have a CME caller, one of the 5 that I've shown you, make calls for these regions. So this says deletion here, duplication. For this region, there's a signal that says there's no call, so there's no change, a duplication here. So we get the same read depth signal, and then we pass it through a recurrent neural network based architecture. We do padding and masking. So we pad each region up to 1,000 base pairs. So each signal at most can be 1,000 base pairs in our application. We do batch normalization. We have a bidirectional LSTM model here, which after batch normalize gives us an encoded read depth signal. Now, after obtaining the read depth signal, we also pass the corresponding prediction of the West-based CME caller here, and then we concatenate them. So we have the embedding of the read depth, a prediction of the base caller, what we call base caller, and then we pass it through 2 fully connected layers, and our model predicts a posterior probability for each region to be a deletion, duplication, or basically no call. So here, for instance, this says a duplication, so the 89% for this region. So it was a deletion called by the base callers, but we say, no, it's supposed to be a duplication. And then the ground truth labels come from the whole genome sequencing CME calls. So hopefully, these are not the ground truth labels, essentially, but the next best thing that we can have. Automated calls made on some other data, which is more reliable. And then we use categorical cross entropy loss, actually a weighted one, to be able to correct these calls. So this is basically the model, the approach to problem. And if you have any questions up to now, maybe I can address them, or then I would go to the results.
We have no questions in the Q&A section. Maybe we can continue, and if there's a question, I will interrupt. Okay.
All right. Okay. So now I will talk about the results of the algorithm. So here you see the duplication precision, deletion precision, and the overall precision, basically, and weighted average of these. So the black bars are the precision after we correct the corresponding algorithm. So this XHMM, the base caller, is at 45% precision. After our correction, it has over 65%. So for conifer, for instance, we even tripled their precision. I think it was around 20.25. It's close to 0.75 right now. For codex, we double it. And as you see in all comparisons, we increase the precision of the three base, the state-of-the-art copy number variant callers after polishing with our approach. And here you see the confusion matrix. This is what we have. The blue boxes are what we have when we don't have polishing. So this is for XHMM, for conifer, for codex 2. So these are the number of duplication events that they call, while this is these show you the ground truths. So dup means if you are on the diagonal, it's a correct prediction by the base caller, and that's what also the ground truth says. But if you are off diagonal, it means the base algorithm makes a mistake. So for instance, this is the corrected version of the unpolished version for XHMM. As you see, we convert like here, the 306 duplication calls of XHMM to no call. So there should be no calls there. And as you see, we keep the number of correct predictions while decreasing the errors. So most of them are passed to here. So you can have, it could have said duplication, we converted to deletion. It could happen, but mostly we get rid of the many of the no calls, erroneous calls. And this is quite striking if you ask me. So this is conifer is quite a conservative tool. It doesn't make many calls. As you see, these are for the averages for the same samples. It hardly makes a call, and you would expect, if it's quite conservative, to be quite accurate, but it's not. And after we correct it with our algorithm, we are still quite selective. We do not basically say all of their predictions are incorrect. We selectively remove them and triple their precision. Here maybe it's a little bit easier because it's quite liberal in terms of making calls, and we eliminate many. But when you come across a very conservative algorithm and still be able to triple the precision of the algorithm, it's quite striking if you ask me. So we were able to improve the precision in all of our benchmarks for these algorithms. So the algorithms that I showed you predict if there's a duplication. It says duplication, but doesn't tell you if you have 5 more copies, 10 more copies. It just says there's a duplication. So these two algorithms here I show you, which is called Control Freak and CMV-Kit, they predict that exact number. They say you expect the copy number to be 2 because you have 2 copies, but this person has 4, 6, 8. So they are making it basically an integer prediction. And we are also able to correct them as well. Now, Control Freak makes 20,000 calls here. And after correcting, if you see that so we basically, so Control Freak says 4 copies, the correct one is 2. So they have an error of 2. And then after correcting with the count, we do the same thing. So now we say the correct copy number should be 6, but the ground truth is 2. So now we have more error, like 4. And we compare our errors, which is more correct, Control Freak as it is, or after correcting with decode. Now, So we have like a curve here. If we are on this side of this line, it means decount is better. So our error is much better compared to control freak. So it predicted like 1,000 copies, but we corrected it. So our error is much better than theirs. So for 20,000 calls, I think around like 12,000 or 13,000, we correct those calls. So we are doing a better job after correcting. And in close to 5,000 calls, or less actually, I think, it should be 3,000 calls, we deteriorate their performance, but by a much smaller margin. Even there is a couple of calls maybe we are doing a worse job. So we show that the correction of decount is improving control freak. So this is the distribution of the copy number calls that they make, and this is the distribution of the ground truth. As you see, they are quite different. After correcting with decount, the distribution of the polished calls is much closer to ground truth. So that's the advantage of decount. And CME kit is a similar algorithm, makes 4,000 calls. As you see, in the majority, we are correcting their calls. And as you see, our distribution is much similar to the ground truth compared to their predictions. So we can also work in the integer copy number, in the integer copy number variation prediction task. So if you have any questions, please stop me, or I can take questions at the end. Now, at the beginning, I said we cannot perform train a model using the human expert labels, but we can use it to compare if we did well. So now, instead of using automated CME calls or whole genome as our ground truth labels, now we are using the human expert labels as our ground truth to check how, check if we are doing a good job in improving the performance of the baseline calls. Again, I have the precisions of the baseline callers. As you see, in all cases, we improve them. So they don't make, for instance, Conifer does not make any predictions here, but still after polishing, the precision increases to 31%. And in all comparisons, the precision of our algorithm was better. But as you see here, the human expert label, when we use human expert labels, the precision is quite low still. I mean, this is the largest one, 0.68. It shows that still there's a gap in terms of calling, automated calling of copy number variations and human experts getting into the process and then doing their job. So there's a gap. And then that's actually the next thing that we are working on top of decount. Being able to have very high precision once you work with human expert data. So this basically shows our performance after polishing, as you see here, once we use a different platform for this sequencing. So we trained our algorithm using data from Illumina Genome Analyzer and Illumina HiSeq 2000, but it's quite possible that your data that you obtain from your sample uses some other technology, even though these are the same. These are also Illumina machines, but has different properties. And then we use so these are actually completely different machines. This is quite possible. You cannot train a model for every technology with decount, but we just check if our performance goes beyond these two technologies. And I think 34 of the 36 tests, we improve the performance, even though this is a different technology.
Dr. Cicek, we have a question in the Q&A.
Okay.
The participant asks, mosaic CMVs are very important in somatically derived human disease such as cancer. Have you tested or plan to test in the future decount for non-integer CMVs, which are the case for mosaic changes?
Okay. We haven't planned that. So why non-integer? I think you mean that a discrete prediction like duplication and deletion. So I mean, we are aware of mosaicism, but we don't have that data about that label about the samples that we have currently. So all we can do at the moment is get the predictions of these baseline callers, CM, XHMM, conifer, or Codex2, and correct them. But if we have some type of an indication, like an input to the model, which would inform the model about this issue, then I think, yes, potentially we should be able to detect them as well. But currently we don't have that data. I hope that answers the question.
Thank you. And we have another participant asking about the model and the data. Is this work based on autoencoders, and what are the dimension sizes of the data?
This work relies on a recurrent neural network, a bidirectional LSTM, to be exact. So the input is 1,000 length vector, which encodes this signal. Basically, numbers indicate the depth of the read depth at each location. And we picked 1,000 here because I think it captured quite a big portion of the copy number variant calls in our data set. So there were larger regions. But when we picked 1,000, it was it captured, I think, 90%. I'm not sure about the number. So we have shorter, of course, but for the shorter calls, we padded and masked them. So each input has 1,000 length in this application. We don't use autoencoders.
Thank you. There are no other questions right now.
Okay. All right. So yeah. So the takeaway message from here is that even if you have the whole exome sequencing data from a different technology, you are able to use our decont model and still be able to improve the precision. So we have our performance generalizes to cross-platform settings. So what if you are using the same technology, but you have a new CMB caller, and for each one of these base callers, we train the decont model. So we have a decont model for XHMM, for conifer, and Codex2. But every day a new algorithm comes out, and then you don't have the resources to train a new decont model for these. Maybe you don't have that many samples. We had 800 samples. Maybe you don't have the power to run that model on 800 samples. Can you use our model? So that's the question. We try to correct the calls of, for instance, conifer using the XHMM model that we trained, or Codex2 using XHMM model that we trained. So this is so if you have green, it means it still increases the performance for DUC precision, DEL precision, overall precision. We observed that the model that we trained with XHMM is the most reliable. Only here, for conifer, it reduces the performance, but mostly, we have 18 tests here. In 10 tests, we had better performance. So we do not recommend this. So it's better to have a caller, a decont model trained for the specific caller that you want to use. But if you don't, we recommend using the decont model that we trained on XHMM to correct your calls. It should still help. That's what our results show. Now, you might ask, even, I talked about it a little bit, but is decont a simple filter? So these callers make some calls, and then we basically change some calls, eliminate some calls. Can I use basically a threshold on the confidence of these calls? And maybe it just serves the same purpose of this deep learning model. Maybe we don't need the deep learning model. So what we did here is we used the confidence threshold on the x-axis you see, and we made it harder for the XHMM algorithm, which is the best one that we had observed in our experiments, to make a call, and made it harder for decont to change a call, in a sense. Okay, so as we go along with the confidence threshold, it's harder and harder for decont to make a call, or for XHMM to make a call. So if decont was a simple filter, you would expect XHMM's performance to crisscross decon's performance. Okay, but once we increase that threshold, decon still dominates XHMM, as you see, in all precision scenarios. And while we increase the threshold, even if it is very conservative, as you see, we still report 1,000 calls, close to 1% precision. So even if decont is very conservative, it outputs 1,000 calls. This is important because what a simple filter would do, as you increase it, it also tends to make less calls, meaning you might end up with no calls with a higher precision. So that's the how to say, the challenge between keeping both high precision and high recall. If you increase precision, you lose from recall. But we see here that decont is still able to make a lot of calls, even though it's really quite sure, from close to 7,000 calls, we are only down to 1,000 calls. You would expect this to be 0 once we increased our confidence threshold. And here, this gap shows that decont is much more than a simple filter. This actually shows our performance across chromosomes. So this is so each dot over here you see is a copy number variation call. And then this is, they are stratified by the human chromosomes. And this is the length of the copy number variation. So there are various versions. The most important is the green one. Here, after correction, it matches the ground truth, while the base correction was not. So these are the XHMM calls corrected by decont. And this shows you the percentage of the calls that are correctly polished. So if you are working with the Y chromosome, you need to polish 84% of the calls, which is quite a lot, actually. Most of them were incorrect by XHMM. We were able to correct them. Or there was one, so on 8 chromosome, it's 80%. So this is the most confident the XHMM was. Even in that case, we had to polish 44% of the calls. So that shows that these 2 chromosomes are quite critical. I mean, that you need to use decont for these. But throughout the genome, decont is useful. And you see green dots over here. You see green dots over there. So it is independent of the length of the copy number variation, showing that even if the base caller has a high variation in their calls, we are able to correct their calls, even if it's not quite variable. So it only reports short calls on this chromosome, we are still able to correct calls. So we are, it's independent of the length of the chromosome or across the chromosomes. So you need decont, Y chromosome and 8 chromosome being quite critical. So I have some results showing that it's not a simple filter. So we don't correlate with some confidence scores. Let's not go into the details. So mappability score, GC content ratio, which you might say maybe decont just looking at these scores. They are not. We don't see any correlation with the embeddings that decont has learned. So it's learning a much more complex relationship between the read depth signal and the copy number variation calls. So these are 2 parameters used by XHMM confidence scores. So if decont was a simple filter, you would expect that decont is only correcting the calls at this corner, which are very low confidence calls by the base caller. But we are even correcting the calls of layers, which are very, very confident. So again, decont is not a simple filter. This is what it's showing. And this is another figure which is showing, so these are the calls by the baseline caller. And these are the ones that are correct. So they are matching here. So it shows that when it is very long, you don't need correction. The baseline algorithm is working well. But what you would expect is like a 500 kilobase calls, which are still long. You need decont correction. Even at the very extreme, you don't. But it doesn't matter if you have long CMEs or short CMEs. You need decont correction. So some other tests. Let me skip this. So highly biological sense. So these are some sanity checks again. let me stop here. So it's almost more than 45 minutes, I guess. So this is my conclusion slide. So we trained a model called Decont to improve the performance of the whole exome sequence-based CMV calling. And we were able to increase the precision by 3 folds in duplication and 2 folds in deletion. So we showed that we are working well in if you are predicting just delts and loops or the exact number of the copy number variants. It's very fast actually, you know, a few seconds per call and then maybe a couple of minutes per person. So you can essentially use it well in a clinical setting. And we showed that our performance generalizes. It's independent of the sequence platform, the capture kit that you use, the algorithm that you use, as we can, you know, polish them no matter what. So Furkan was a master's student in my group. He's now pursuing a PhD at Oxford University, and Can Alkan is a peer at Decont here. So this was a collaboration with them. Thank you for your time. Again, our paper is here, the code is here, the data is here, and I'm happy to take questions. Thank you.
Thank you for the nice presentation and congratulations on the impressive work. We have a question in the Q&A section. Is the code running on GPU? The participants asked.
Yeah, so we used 4 GPUs here, a Titan, which has 24 gigs memory, and then 3 2080s. So in 4 GPUs in total to train the models. I think the largest model that we trained took 4 days, I think. So, yeah.
I had a question before the, I prepared the question before the presentation. I haven't read the paper in the presentation, but I found the thesis by Mr. Özdan, and he states the training hours, total training hours ranged from 20 hours and 95 hours, like you said. And I also checked the GitHub repository of the project. How did you distribute the training to multiple GPUs in terms of technologies? I believe you have used TensorFlow, and any training strategies, any tips you can give to the audience?
I think we use, so let me remember, we are using, I mean, in my lab, so the students are kind of free to use any technology. I think we used, from the start, we used a lot of PyTorch. Now it's more Keras. I think this was on Keras, if I remember correctly. Just use CUDA to distribute the process. Nothing in specific, actually. Pretty standard procedures. But it wouldn't fit to a single GPU, so we had to. I remember that. So it was. I think one of the. I think the Codex 2 model was quite challenging because it's making a lot of calls as opposed to Conifer, and then we had to spend a little bit of time and also use GPU memory to scale.
Okay, so any plans on, for example, benchmarks to obtain performance numbers for multiple GPU settings, like how the performance scales on multiple GPUs? And I was going to also ask, can scaling out in an HPC infrastructure to multiple nodes and multiple GPUs can provide some benefits to your work, to your overall goal?
Yeah. So usually I mean, this would, for instance, be much more relevant for John's work. He's more into sequencing. Usually the work that I do, even if we have these large models, these are trained offline and then I have to use them once. And even if I have 4 days, since the model is trained only once, I don't need to update it frequently. It's not a big challenge. And in bioinformatics, unlike your domain, I know, it is not quite relevant to scale onto multiple GPUs, showing the time performance increase. Usually people even ignore them. You should have seen that we have maybe a single paragraph that talks about the time requirement, how many GPUs, because nobody really cares about it in our domain since it's trained once. So I don't think we'll go to benchmark this, but if this scale of the work kind of goes higher, that we have to train many more models with much more data, which I don't see at this point, then we would certainly need HPC for this purpose.
Okay, I was asking because of the EuroHPC relevance that we have talked about.
Yes.
Let's wait for maybe 1 more minute if there are more questions. Maybe I have one more question. What about the data preprocessing step? In the thesis, I also read about the data preprocessing step. Was there any need to distribute data preprocessing over multiple cores or accelerators, use accelerators?
Let me think. So I remember it was a painful process. Usually, like people say, more than half of bioinformatics is data crunching. So we had to run all these algorithms, all these 5-6 algorithms, baselines on the raw data. I do not remember that need, since none of them are. These are certainly not machine learning algorithms. We didn't use any GPUs, but I remember we used parallelism on a 40-core machine. We didn't distribute XOR servers, I guess.
Okay, thank you. There is a question on the Q&A. Any plans on tumor normal calls?
That's actually a good question. This works on germline, which I kind of skipped talking about. So this is basically a healthy person just goes and then you find their copy number variations. One other application of this, is you get a sample from a tumor and a sample from the normal cell of the person and then compare the signals, which is quite valuable. So what we had done, actually, there's a section in the paper about this. We checked if we are able to correct the calls on the cancer sample. So these base callers, some of them can take both normal and then cancer tissue as input. So we ran them like that and then corrected them. Mostly they didn't make any calls. So once they do not make any calls, we could not polish them, and we don't have anything to do. But then we tried, in one sample, I believe, they reported a duplication on the cancer sample and a deletion on the normal sample, or vice versa. I don't remember which one. But this is quite problematic, which is very unlikely to happen. You would have a normal one, a duplication or a deletion. One would just vary. You don't have the normal vary the one way and then the cancers vary the other way, which is unlikely. And we showed that we corrected the normal sample. Currently, so we are looking for a follow-up on this to build a standalone, not a corrector, but just an algorithm which predicts these calls using the same idea. And there we might actually incorporate this idea by basically initially checking, maybe pinpointing the regions which are different between the tumor and normal. And that's a good question, actually. We are looking at that. Thank you.
Thank you. Let's wait for a couple of more seconds, maybe more questions. Okay, thank you again for your presentation, and
thank you very much.
Thank you for the participants for joining us today.
Yeah, thank you very much.
Goodbye.
Yeah, thank you. Bye-bye.
Bye-bye.